So proteins can be looked at in levels. If we get really, really close, the bottom line, the very first level of structure that we would see is called the primary structure. So this would be like looking at us on the chemical level. The primary structure is the order of the amino acids. Now, I don't know if you remember, but every amino acid is identical except for one thing. You remember what the one difference is?
The R group.
The R group, good. So on one side, we have this, which is called, does anybody remember? What's the first part of the name of this?
Amino.
Amino. So this is our amino group. And on this side, correct, we have the acid, which is actually a carboxyl or carboxylic acid. And that's where our amino acid comes from. And then we have a hydrogen, and then this is our R group here. So that's the one thing that differs. So if we were going to describe a protein on the primary level, we might say this particular protein is 120 amino acids long. And the amino acids are in this order: leucine, glycine, valine, blah, blah, blah. They have names. In this diagram, instead of showing the letter R, they're actually showing you some of the R groups. So if we look here, here's an R group, here's an R group, an R group, an R group. And so it looks a lot more complicated, but all they're really doing is they're replacing the letter R with what really goes there. And that's what's going to determine what that particular amino acid is. Now, the order of amino acids is going to determine everything else about how that protein behaves. Remember, DNA codes for making these proteins. So the DNA at some point coded to put these amino acids in this order on this particular protein. Yes?
How do you know which one's the R group?
Oh, well, I know that one of them is always a hydrogen. So if I eliminate the hydrogen here, here's the hydrogen, the hydrogen, the hydrogen. The other one has to be the R group. So you always have your amino, carboxyl. Huh?
On the carbon?
On the center carbon. Now the other thing is about this, if you were looking at a drawing like this that was so complicated and you don't see the letter R, one way you could recognize that it's a protein chain is look for the repeating NCC structure. Here's your NCC. That would be one amino acid. NCC. Here's a second amino acid. So you can ignore the complex stuff sticking off of it if you see that backbone, because that doesn't change. No matter what amino acid it is, you're still going to have that repeating structure.
NCC is just one, right?
NCC would be one, right? And remember what happened here is here's the one on this end. Here's our NCC, and then here's our second one, our NCC.
I say Maria Bastera.
Yeah, Maggie. Now you'll be recorded for posterity in my recording. All right. And then here we have our amino group. So here's what happened. In order to connect them in this peptide bond, this connected with this, and they left. What did they make?
Water.
They made water. And what do we call that reaction that put them together?
Synthesis.
A synthesis. It's actually called, remember, what happens when you lose water?
Dehydration.
You get dehydrated, so it's dehydration synthesis. Okay, so that's our primary structure. Now we're going to go to the next level. If we were to back up, we would see that in sections of this chain, there would be folding. And there's two main kinds of folding that we see. One of them is called an alpha helix, which looks like this. It's kind of an actual helix. Sometimes it's written with the Greek letter alpha, just so you know. If you see that funny looking fish helix, that's alpha helix. And the other pattern that we sometimes see is called a pleated sheet, or sometimes it's actually called a beta pleated sheet. Now, these patterns come from the reactions of the hydrogen on one amino acid with the oxygen on another. So here's our nitrogen, the amino group on one, and here's the carboxylic acid group on another. And because they're attracted to each other, we now get these patterns. So this is basically taking a step back and saying, because of the amino acid order, we're now seeing a folding pattern. The alpha helix is pretty easy to visualize. Most of you know what a helix is, sort of like taking hair and making like a curly cube. The pleated sheet's a little harder to visualize, but imagine that because of attractions between this chain and this chain, it causes them to kind of fold up. This is what they mean by accordion style, that they're attracted, that this one is attracted to this one. So this would be one protein. This would just be a section of the protein where they were attracted to each other. So that's called secondary structure. The attractions of hydrogen bonds basically between the amino group of one and the carboxyl group of another. All right. If we were to back up even further, we would see that this entire protein folds up into what looks like a glob, but it's not. It's a specific shape. And that's called tertiary structure. It's how the entire protein folds up into a final shape. And that shape comes from disulfide bridges. Basically, this is R groups reacting with each other. You're actually getting ionic bonds between them. You're actually getting what are called disulfide bridges. If you go back to functional groups that I'm sure you all are learning, disulfide bridges are reactions between one sulfhydryl or thiol group with another one, and they form a bridge. So here's a protein, and this is the whole chain, and it's folding up into a particular shape. Like I said, it looks like a blob. It looks like it doesn't really have any rhyme or reason, but it does. I don't know if anybody remembers learning about enzymes. And when you learned about enzymes, is that they speed up reactions, and they were kind of drawn probably like this. And that was the enzyme, and this was the substrate. And the enzyme had this area called the active site. Does that sound familiar at all?
Yes.
Yes, to some of you? Okay. And I drew it looking like Pac-Man when I taught it. Yes. But the fact of the matter is an enzyme doesn't look like Pac-Man. We draw it that way to make it more easy to visualize what an enzyme looks like. This is what an enzyme would really look like. And that active site would really be a crevice like this created because of the way this folded up. This would actually be what an active site would really look like. But it's much more complex and crazy looking to draw all this out. So once we're on the level of talking about enzymes, we simplified it. Just like when we were talking about the cell membrane, we don't draw out the phospholipid. We just represent it with a little circle and two sticks to simplify. Well, they're actually like this because they're unsaturated. So, all right, so that's tertiary structure. The last level, if we were to back all the way up, some proteins, not all of them, some proteins only go up to tertiary, but sometimes the final functional protein is actually consisting of more than one chain. And that's called quaternary structure. That's the biggest structure. So, for example, this is hemoglobin on the left. You can see it better on the TV. But if you look at hemoglobin, the final functioning hemoglobin is actually composed of 4 separate chains. So here's 1, there's 2, here's the 3rd one, and a 4th one here. So the final functioning hemoglobin is actually made out of 4 chains, which all have primary, secondary, tertiary, and then they combine into the final structure, and that's what hemoglobin looks like. This one here, and you don't have to know these in particular, this one here is how insulin is stored. It's stored as a hexamer. How many do you think are in it? 6. Hex has 6. So in this case, this storage protein consists of 6 different chains, all combined. And this one I thought it was kind of interesting. This is HIV protease. This is a particular enzyme that HIV needs in order to make copies of itself when it infects you. So one of the ways that HIV drugs work, anti-HIV drugs, I should say, you don't want to take drugs to get HIV, is that it inhibits this enzyme so that the HIV can't make copies of itself. And so basically you've shut the virus down by targeting a specific protein enzyme.
Do you have that enzyme in you, or does it come?
No, what happens is the virus, when it infects you, it injects, in this case it's RNA, but basically it's nucleic acid. Your body follows the instructions. So then your cell reads that nucleic acid and manufactures the enzyme. So by inhibiting the function of that enzyme, it's not an enzyme you would normally be using. It's an enzyme that only is needed to build more viruses. So we can inhibit that enzyme. All right, and this last slide is just showing you, hopefully, kind of a summary of the whole thing. So if we were to go in with a microscope and look at the closest, most simple level, this would be our primary structure. Here they just wrote out the names of the amino acids. Depending on the R groups of these amino acids, it's going to determine how this thing is going to fold up. the first thing that's going to happen is little areas are going to form repeating patterns. And that's our secondary structure here. In this one, it's an alpha helix. In this little area, there might be a pleated sheet. Oops, I didn't mean to do that. There might be a pleated sheet in another area over here. This could have a pleated sheet. And then this is tertiary, so now this is the whole thing. And then in this particular case, they're showing quaternary because they're showing that this final protein actually consists of 4. All right, so now that probably made a little bit of sense. But